Hello, my name is Peter Fedorov. I'm head of the Neurophysiology Research Group at the University of Innsbruck. The purpose of this video is to present to you the PManalyzer. That's a MATLAB-based software tool that we use in our group to analyze human movements. The PManalyzer was coded by Thomas Hyde as part of his PhD project and is published in Frontiers in Neuroinformatics. For a quick reference, you have the DOI number down here on the screen. If you want to analyze human movement data, obviously the first step is to have human movement data. Now, for the PManalyzer to be able to read it in, you need to store your data in the same folder. You can analyze several subjects or data from different trials, but they have to be in the same folder. And you should use the following ending for your file name. The file name should end in S, small s, and then a number, and small t, and then a number. These letters stand for subject, and then the subject number, and trial, and then the trial number. So here in this example, you can see we have 3 subjects, and from every subject we have 8 different trials. It only makes sense to analyze movements from different trials or different subjects, obviously, if they performed the same movement. I do not recommend to mix walking and running data, for example. The file extension needs to indicate an ASCII format, so at the moment you can read in CSV data as we have here, or TXT data. Both are data formats that typical data recording systems like Vicon or Qualisys can export. The software, the MATLAB codes for the PManalyzer, can be stored in a different folder. So what you see here is what you find when you unpack the PManalyzer as downloaded from either the paper or from the software webpage that our group has created. In order to start the PManalyzer, you need to find this file here, principalmovementanalyzer.m. If you double click that and you have MATLAB installed, then the PManalyzer should open and become ready to use. If you double click the Principal Movement Analyzer, then this general user interface opens here. So we have MATLAB in the background and here a separate window for the control of the Principal Movement Analyzer. This control looks pretty busy, but in principle there are 4 fields. The first field here is input data. The second field controls the computation and what output is generated. Then we have a field that allows you to select plot options and a field that allows you to select video options. Down here we have the option of saving or loading settings. That means the settings of this general user interface that you see in front of you. And here in the middle you have a big start button, so once all the settings are set correctly, you can start the computation by clicking here. Let's go through the fields one by one. Here in the input data, the first step is to copy in the path where your data is. So this is the path to the folder where the data is. That's the folder I just showed you. Here in the subject trials, here you can select how many subjects and how many trials you want to analyze. You have seen in the folder we had 3 subjects and 8 trials. However, here I can select to only run the analysis on 2 subjects and only on the first trial. I can also specify a description of what analysis I'm currently doing. That description is then saved with the output information. The second step is to specify what type of data we want to read in. So here you have different options. For example, we have CSV files, as you saw just a few seconds ago, and then there's the option of, for example, deleting headlines, for specifying delimiters in the file. So what separates the data from the next data point. The sample frequency should be specified here, and you can also put in the number of markers and the number of rows that are expected. These are not essential information here. Should the number of rows, for example, differ from what you put in here, then MATLAB will generate a warning message but continue with the program. You also have the option of some preliminary data treatment. For example, you could delete some columns or some rows. For example, if you want to cut off the end of the file or a few seconds of the beginning of each file, this could be done here. The delete columns allows you, for example, to delete columns that have timestamps on it or frame numbers in it. You can, of course, also delete columns if the data of one marker is problematic and you want to exclude a marker from your analysis. This could be done either by deleting columns, or you give the number of the marker within the matrix to delete here. Sometimes human movement data have gaps in recording if the marker is not visible at certain time points. You can either use the gap filling options that other analysis, for example the motion analysis, motion recording software provides. We have integrated a gap filling program. It's also a MATLAB based program that you can select here should there be gaps in your data set. If there are no gaps, of course you have the option to use no gap filling. So for the moment I recommend the setting here, every data set separately. Should MATLAB find a gap, then it will fill it. Are there no gaps in it, then nothing will happen. You can also perform coordinate transformations or filter the read-in data. At the moment, I don't recommend to do that as a first step. The second field here now performs normalization. In order to analyze data from different trials, and even more importantly, when you analyze data from different volunteers, then some normalizations are necessary. And some normalizations are done automatically. For example, that the mean posture is subtracted from each trial data. That is done automatically. You also have the option of performing additional normalization steps. One step we often use is the normalization to mean Euclidean distance. That's a setting you can select here, but there are also a few other types of normalization available. Furthermore, you can weigh the markers, for example, with the relative weight that a marker represents in your data. However, these weights you have to specify. So this only works if you have defined in the code a weight vector, which is something that we'll show you in a different video how to do that. Finally, when you want to analyze the principal position, principal velocity, and principal acceleration, before you differentiate your data, it makes sense to filter to low pass filter your data. And so what we have as a standard implemented here are Butterworth low pass filters, where you can specify the order and the cutoff of the filter. Additional options that you can set here are cross-validation, or that a power spectrum density is calculated. For the first trial, I recommend to not click these options here. This defines what is done before calculating the PCA. The next field here defines what output is calculated. So you can define here the name by which the output is saved. For example, data PCA here. Or you can, if you want to read in data, so you don't want to read it file by file, but you have already run a principal component analysis and you have saved some old data that you want to reanalyze, then you can also, instead of perform computations, new computations, you can load in data. That's an additional option here, but not further interesting for the moment. So at the moment I recommend these settings. Another setting that is interesting would be to click these 2 points. These are specific if you are interested in, or if you're analyzing postural control data. Then a separate Excel data file is created as output, which contains several variables that are calculated from the principal movements that the PCA outputs. That can be used then to perform statistics on it. So the data file that is created in the Excel can be, for example, then imported into SPSS or other statistics software. The third bigger field here is the plot options. You can select to create plots or not plots. So for a first run through, I'm showing you here only a few of the plot options. You can also define here how you want the plots to be saved. Maybe only as JPEG or as JPEG and MATLAB figures. MATLAB figures obviously allows you to further edit the plots after they have been created. That's not really possible if you save them as a JPEG form, but of course JPEG will allow you a much faster run through the processing. So for this video I'm selecting JPEG here, and here I'm showing you output of eigenvalues, overall eigenvalues for every volunteer. Then we can go to the video options. Again, you can turn that off, videos disable. You can have 2D options, which I will show you in this video. Also 3D videos are available as an option, but I will also move that into a different video. The video settings that I recommend here are that here you can specify an extra folder where the videos will be stored. And then you can select what volunteers you want a video to be created for. So here I selected the second volunteer. Be sure, of course, that you select somebody that is in your data set also. So here we have included the volunteers 1 and 2 in the analysis, and we can select 1 and 2 here. I selected 2. Same for trials. We included trial 1 here, and I will display off volunteer 2 the first trial with videos 1 to 4 in the same figure. So one figure will be created, or one video file, where we will find 4 principal movements being displayed. And here we can select the output trial. So this option here means that a video is created from the actual trial as it is read in. We have additional options. For example, up here I checked input data post-processing, so that shows after some of the normalization steps the input data, the original movement as recorded. And then we have here PM visualization. This PM visualization shows what how the movement components look like without showing the actual time, the actual behavior. It is only a sweep from minimum to maximum that was observed if I select this here. So this allows a very fast idea of what the movement represents, but it does not represent the accurate timeline that we can observe. For the moment I will only use this one, so I will show you specifically the second subject and the first trial, how that person moved. I have the option of different axis settings, as you see here. So if what the right setting is here, that depends on how your data collection was calibrated and what axes were defined by the system that measured the motion data. But you have here different versions available. So if your videos look very strange, maybe switch from a different axis setting here. Furthermore, we have the option of amplifying the movement amplitude. So the videos that I create here are videos of postural control data. That means our volunteers don't move a lot, and if we split that very small amplitude movement then into different movement components, then we get even smaller movement amplitudes. So in order to see anything at all in these videos, we need an amplification which can be set here. So the first two videos will be amplified here by a factor 15 and the second two by 30. So you can either here put in two numbers, or maybe more precise would be to specify for every video that you create the specific amplification factor. So we will create videos, or one video of 4 movement components, and the first two will be amplified with 15, with a factor 15, and the second two with a factor 30. Skip frame allows you to not display in the video every frame, but to jump to a frame a bit later. So in our case, we skipped 25, so we will make jumps of 25 frames. That means the video will run a bit faster. We don't have to wait very long for the video to be created. These settings we can save, for example, as test here. So if we want to use them once more, then we can load them in later. So I have saved them now, as you saw, with test setting, and you saw there that some warnings will appear if you are overriding a previously stored setting. Now we are ready to start the computation. If I click here, you see in the background that MATLAB starts to work. It's a good idea to keep an eye on these MATLAB outputs. If you see the red lines indicating error messages, then something went wrong. Maybe there were gaps in your data and you didn't use the gap filling, or there could be other settings that were wrong. Maybe you selected subjects that are not in the data folder that you specified. That would produce error messages that you find here. As long as you don't get any error messages, then in the background the software is running. And maybe a warning, we have here in this case only 2 subjects, only 1 trial. And you see still the processing and the generating of the output takes quite some time. Obviously that takes much longer if you have more data, so be patient here. So you saw in the background that some of the plots, output plots, were created, and they are automatically stored and then closed again. And we have already seen now the video of the input data, and what you see here is the output data. So we selected 15 as amplification factors for the first two and 30 then for the second. So we can actually see some movement in this person here, as you see, standing bipedally on a force plate, and that was the movement showing you the actual movements split into the first 4 movement components. We get a number of messages here. It's always good to find this message here, GUI computation finished successfully. So that means your PManalyzer completed successfully without error message the calculation, and after you click that away, the tool here will be available for another calculation, maybe with another setting if you want that. You will find the output that is generated in the data folder that you specified earlier. So I have to go back to the folder where I have my data. Here now an output folder was created. So it provides some information here, for example, about how the data was filtered. If you double click this, you find 3 folders. One with the data, so that contains the MATLAB data. If you want to program yourself some post-processing, this is available here. If you go back, if you are looking for plots that you want to generate it, then you find that here. These score plots I didn't select in the calculation, so this it's not empty because there is something from a previous run through, but what we calculated here are these graphs that you may recognize that they briefly showed up when I executed the PManalyzer. So here you see, for example, the eigenvalues, or here actually the eigen standard deviation, here the eigenvalues, the variances that were calculated, or cumulative eigenvalues. Here another form of showing the eigenvalues. So the plots are stored here. And finally, videos are created here. So the videos we created in this run through is the original motion. So this would be an example of the video. Here no amplification is used, so I'm not sure if you are able to see any movement at all, since that's a quiet standing trial. The person was not moving a lot. Then we have the subject visualization and the PM visualization. This PM visualization is what I explained, not the actual timeline, just a sweep through from minimum to maximum amplitude. And this one are videos that are created from the actual person, the actual volunteer that we analyzed. So we had selected subject 2, trial 1 for creating videos, and we had selected the first 4 videos and selected 4 videos in the same video file. So here you see the movement, the first 4 principal movement components that were calculated in the run-through that we just performed. So I hope that gave you a first impression of what the PManalyzer is and how to perform a first analysis of data. We will create more videos that will allow you, for example, to create better videos. At the moment you see only here the marker positions as they were read in. Obviously it looks nicer when you link those marker positions so that you get nice stick figures. You can also change the color, and there are other options that will be explained in a separate video, and we will also create a few more videos with looking at other options, looking at how to interpret data that is or output variables that are calculated, but that I will keep for other videos. For the moment, thank you for your attention, and I hope that gave you a good impression for what you can do with the Principal Movement Analyzer software. Thank you.